This butterfly is trying to find some food, but it only wants one kind of flower. Before it gets there, the butterfly must navigate past the sight and scent of other flowers. In a complex sensory landscape such as this, how do pollinators find their plants? Mutualistic relationships like pollination are found everywhere in nature and are responsible for sustaining much of the biodiversity central to our ecosystems. Therefore, understanding the mechanisms behind the origins, maintenance, and potential loss of these mutualisms is critical. Pollinators utilize a wide range of visual cues and chemical signals to locate their host plants. But these systems are often more complex, which makes it difficult to study these interactions in depth, especially when looking at multiple signals. That's why I'm excited to use cycads. Cycads are an ancient extant lineage of gymnosperms at least 200 million years old that evolved before flowering plants. Cycads are also dioecious, meaning that they have separate male and female or pollen and ovulate plants. They also have obligate brood site pollinating weevils that spend their entire lives living in the male cone. Zamia integrifolia, the cycad I'm studying, is also thermogenic, meaning that it generates its own heat. Byproducts of this phenomenon are increased scent, humidity, and carbon dioxide. The intensity and timing of these signals is thought to push the weevil from the male cones into the female cone, effectively pollinating the plant. This is important and exciting for others because cycads are endangered, and I will be working with a conservation garden to study how multimodal pollination signals maintain this mutualism, and how they are contributing to an observed host shift, possibly threatening that conservation. To study this, I will continue collecting temperature, humidity, and CO2 from cones the weevils visit. I will also collect scent using headspace collection, and I will use a GCMS to analyze compound production throughout the day. I hypothesize the weevils use these signals to inform their host choices, and will test this by using an electroantennogram. This will elucidate their response to different intensities of signals and test the physiological limits of their detection. To determine the direction of the weevil's response to the stimuli, I will use a full factorial design and expose the weevils to individual and combined signals in a Y-tube and olfactometer and measure their behavioral response. I will also compare these responses to behaviors measured in the field. I hypothesize the weevils shifting hosts in the garden perceive signals differently than those in their native range. I will compare naive insect perception of signals, their behavioral responses, and will use genomic resequencing and fixation index analysis to determine if they are evolving at a faster rate than those in the garden. By researching the signals involved in the evolution of plant-insect interactions through the lens of a unique and ancient obligate pollination mutualism, these results will provide insights into the traits that contribute to the maintenance and potential breakdown of mutualism specificity. It will also contribute to the conservation of endangered cycads through a better understanding of their mutualistic relationships.